Right, my very dear students, today we are going to talk about a very important aspect of endocrine system, that is about the thyroid system, right? Thyroid hormone and thyroid gland. Thyroid hormones. Now, thyroid hormone, they are anabolic hormones or catabolic hormones? Yes, doctor. The anabolic hormone or they are catabolic hormone dominantly when they are present in the body in normal level. Yeah, they are anabolic, right? Normally, but of course when this excess of thyroid hormone is present, it may lead to excess catabolism. Anyway, first of all, we must know what are the major functions of, what is the most important thing about the thyroid hormone is the thyroid hormones are concerned with mental development, mental growth, maturation, physical growth and maturation, basal metabolic rate, and they also maintain sensitivity to sensitivity to adrenergic system, adrenergic system, right? We'll go into detail of all these things, that how it lead to mental maturation, how it lead to physical maturation, how it is maintaining our basal metabolic rate, how it is keeping our tissue appropriately sensitive to adrenergic drive. These things we'll be discussing later. First of all, we'll talk about the structure of the gland very briefly, right? And follicles related in that, right? Now, you know that basically thyroid hormones are produced by the thyroid gland, and thyroid gland is present in the neck in its interior part, just in front of the, just below the larynx, in front of the trachea, right? And thyroid gland is normally a bilobed structure, right? And in this bilobed structure there are two upper pole and two lower pole, and there's a middle isthmus binding the both areas. Like other tissues in our body, thyroid gland has a lot of stroma, right? And its functional tissue, which is a truly functional unit of this gland, that is called follicle. Let me tell you what are the follicles and how do they work, right? Let's look very briefly, very briefly at the structure of thyroid gland. Basically, thyroid gland has these very specialized, you can say, functional unit, which are made of cuboidal cells, and these functional units are basically made of epithelial cells. These epithelial cells make a spherical, you can say, structure which is called follicle. The structure is called follicle, right? And there's too many follicles. There are many follicles in one person's thyroid gland. So now we can say what are follicles. Follicles are specialized structures, right? These are made of thyrocytes, so thyroid functional glands, and these follicles are spherical structures. In cut section they look like circle of epithelial, you can say epithelial cells, usually cuboidal cells, and these follicles are having in between stroma, especially let's suppose this is a blood supply, capillary network of thyroid structure. Here is also another capillary. And another important thing which is there, that in between the capillaries and follicles, in some areas you find a very unique type of cluster of cells, and these blue cells are called parafollicular cells, because these blue cells are on the side of the follicles. So these blue cells are called parafollicular cells. These parafollicular cells are responsible to produce calcitonin. Calcitonin, a calcium lowering hormone. And follicles, when they are functional, they are responsible to produce T3 and T4, which are thyroid hormones. So we can say from the thyroid gland there are two types of hormones coming out, T3 and T4, which are derived from the follicles, and calcitonin, which is derived from the perifollicular cells. Is that right? Now, follicles are basically responsible for T3 T4 secretion, and again I'm repeating, parafollicular cells are also called C cells, C for calcitonin. They are responsible for calcitonin handling. T3 T4, they act on every cell in the body. T3, T4, they act on every cell in the body because they're responsible to maintain the metabolism throughout the body. And T3, T4 especially are important for us to mental development and maturation, as well as they are especially important for physical growth as well, right? Moreover, T3, T4, they also keep the tissues sensitive to adrenergic drive, right? Opposite to that, when we talk about calcitonin, calcitonin is basically a calcium lowering hormone. In today's session, we'll be mainly concentrating on thyroid hormones, that is about T3 and T4, right? We'll talk about first of all that what is the basic structure of a thyrocyte, right? Thyrocyte means a single cell which is contributing to a thyroid follicle, and how a particular thyrocyte is collecting the raw material and synthesizing, yes please, synthesizing thyroid hormones, how it stores the hormones in colloid, and eventually how from colloid T3, T4 are absorbed by these cells and secreted into blood, right? So let's go into detail of one follicle, that one single follicle, how does it work, and what is the particular role of a one single thyroid cell or thyrocyte. First of all, we'll talk about how T3, T4 is synthesized and released. Let's suppose I draw one follicle over here, right? As I told you, one thyroid gland has many follicles. Follicles are the basic functional units of thyroid gland concerned with the synthesis of T3, T4. Now, this is a follicle of a normal thyroid gland. As you know, this follicle is made of what type of cells? Cuboidal cells having a nucleus in the center. Now, you know that capillary system is an outer aspect of this, right? Blood flow is around the follicle, right? And this membrane of follicle, this membrane of a follicular cell, right? This membrane is called basal membrane, and this membrane is called, yes please, lateral membrane. Here is also a lateral membrane, and at this point, this part of the follicular cell membrane is called apical membrane, or this is also called luminal membrane because it is facing to the lumen of follicle. The center of the follicle, this area is basically lumen of the follicle. Again, let me repeat it. If I draw this single cell, another cell, right, with different colors, now this part is which one? This is facing on the lumen, right? Here it is, yes. Which part of the cell? Lateral. And what is this part of the membrane? Basal. Is that right? Now we have to see what are the basic principles involved in synthesis of and synthesis, storage and release of thyroid hormone. Step number one is that these cells have to take the raw material from blood side. It means it's the basal side of the, yes, it is the basal membrane which is collecting the raw material from the capillary side or interstitial fluid. So step number one should be in synthesis that it should collect the raw material. Then step number two should be that within the follicular cell, within the follicular cell, there should be synthesis of thyroglobin, right? Raw material like iodide we have taken from the blood. Then we synthesize the thyroglobin. Then step number three is release of thyroglobin into where? In the lumen. There certain important synthetic steps occur which will go into detail. Then there is a storage phase, that thyroglobin and its associated T3T4 are stored within this luminal area. And this luminal area, when it is full of thyroglobin, that thyroglobin is called colloid. That is called colloid. It means this is the colloid which is secreted by every cell. And this colloid is what? Colloid is basically, yeah, what is this colloid? Thyroglobin, right? And as a part of thyroglobin, there is synthesis of T3T4. Then again, listen. Step number one: take the raw material from the interstitial area into the cell, and this function is present into which part of the membrane? Basal membrane. Then within the cell, you synthesize thyroglobin, right? Then thyroglobin and iodides, they are thrown through the apical membrane or luminal membrane to the colloidal area, right? Luminal area, where with some synthetic important steps are done, and then T3T4 as a part of thyroglobin is stored within the colloid. And when we need T3T4 in the blood, of course, if we need T3T4 in the blood or for the peripheral tissue, we have to take from the colloid to back to the blood. If we have to take it from the colloid back to the blood, then it has to be taken, yeah, to the cells back by the endocytic process. From here, we will make the T3T4 pull out of the thyroglobin structure, and eventually T3T4 should come into circulation. So it means how many steps are there? There's 7 steps in synthesis, storage, and release of T3, T4. Let's remove one cell and go into detail of the function of this one particular cell. I'm going to remove this cell and put it over there and go into detail of its function. Let's suppose this is the basal membrane, base of the cell, right? This is its which part? Very good, lateral. And what is it here? Apical part or luminal part. Is that right? Now, I've removed this cell over there and we'll see how it is functioning. And of course, as good student, you won't forget that there are other cells also on the side of it, right? For example, I can make other cells just to show you, so that now yeah, now these are follicular cells, and these follicular cells have central area in which colloid has to be collected. And here is your circulation. Here is your circulation in thyroid gland. Now we'll concentrate on the function of this particular cell. For synthetic purposes, it needs raw material. One of the most important raw material which is needed by the cell is iodides. You know, iodides are coming from GIT. Iodides are normally present in our diet. From there they appear into our circulation. Iodides appear into circulation, and of course from our circulation they spread all over the body and into interstitial area. Remember one thing, all the iodides which are daily absorbed from the GIT, about 20% is usually taken up by the thyroid gland or thyroid follicular cells. 80% of these iodides are lost into urine. Again, let me repeat it. The iodides is normally present abundantly in our diet, but in some areas it may not be present in sufficient amount. That is why in many parts of the world table salt is iodized, in which one part of iodine is present in 100,000 part of the salt, right? So that there should not be deficient supply of iodide to the population, right? Let's suppose if you are taking enough amount of iodides in your food, iodide will be absorbed from GIT in the same way as chlorides are absorbed, and eventually iodides will appear in your circulatory system. Out of circulatory system, about 20% of the whatever iodides are present in circulation daily, whatever iodides are coming from GIT to circulatory system daily, about 20% is taken up normally by the thyroid tissue. Which part of the thyroid tissue? Follicular cells, right? And 80%, you can say 20% is going to the thyroid tissue, and about 80% will eventually go to the renal system, and from there it will get lost into urine. Is that right? Now, this iodide, how it will come into cell? You know, iodide is basically charged molecule, and charged molecule or polar compound cannot dissolve into lipid, so they cannot enter directly into cell. So iodide has to be taken by a special mechanism from the interstitial area into the cell. For that purpose, there is a very special pump over here, the very special pump over here. And this pump, right, this is active transport mechanism. We also call it iodide trapper or iodide pump. These iodide pumps are specially present in basal membrane. Iodide pumps are not present on the lateral membranes. Iodide pumps are not present on the apical membrane facing the lumen. Iodide pumps are present where? Again, will you help me to remind it? Yes? Yeah, iodide pumps are present on the basal membrane, right? Because they have to take iodides from the circulation or interstitial area into the cell. As I already told you, that iodides are basically charged molecule. They cannot dissolve. So they have to be taken into the cell, right? And when they enter into the cell, they are taken by active transport mechanism. They are taken up by, yes, active transport mechanism, and iodides are concentrated into the follicular cells against the concentration gradient and even against the electrical gradient. As you know, these cells do have resting membrane potential. These cells are also electrically negative inside, right? So they, if negative ions have to be pushed into cell, they are moving against the electrical gradient. In the same way, iodide is less concentration in the blood and higher concentration into these cells. So it means they are concentrated. Yeah, they are concentrated into the cell against the electrical and chemical concentration gradient. Once the iodides are present within the cell, right, they will diffuse down through the cell towards the luminal side. Iodides will move towards the luminal side. When iodides are moving towards the luminal side, let's suppose these are iodides, they are moving towards the luminal side, right? There is something extremely important, that there is an enzyme present over here, and this enzyme, yeah, what is the name of this enzyme? Yes, what is the name of this enzyme? Okay, let me tell you the duty of this enzyme first. The duty of this enzyme is that as iodides are passing through this membrane, when they appear here, they are converted into iodine. They are converted into iodine. Is that right? And in this process, they use hydrogen peroxide. It means this enzyme is leading to oxidation of iodides. Iodides are oxidized and converted into iodine. So what is the name of this enzyme? This enzyme is called peroxidases. The name of this enzyme is called peroxidases. Okay, let me tell you what are the different functions of peroxidases. Again, peroxidases are special type of enzymes. Right. So we were talking about a very important enzyme, that is thyroid peroxidase. Now, thyroid peroxidase has many functions. One function of thyroid peroxidase was that it was converting, yes, it was converting iodides and oxidizing them into iodine. It was converting them and oxidizing them into iodine. Is that right? This was one function of this enzyme. Now, this enzyme is basically uh, present and attached with the apical membrane, right? And because it is attached with the apical membrane, right, it has very unique way of functioning. This enzyme has a very unique way of functioning. Number one, yes, number one, it is leading to this function number one. What is the function number one? It is leading to oxidation of oxidation of iodides into iodine. This is one function, but it has some other functions also. For to discuss those functions, we have to come back to the cell, right? Cell is also involved into one more function, that is the formation of unique type of protein called thyroglobin. Again, how thyroglobin is formed? It's too easy to understand that here is the nucleus, and in the nucleus, you know, there are genes, right? In the thyroid cells, a special type of gene is activated, and when this gene is activated, this produces a messenger RNA, and this messenger RNA, when it is translated, of course, through the ribosomes, again, there is a unique type of gene present over here, and when this unique type of gene expresses itself, it makes a unique type of messenger RNA. And this messenger RNA, when it is translated, it makes a unique type of protein. And this protein, which is synthesized by this messenger RNA, with the guidance of this messenger RNA, this protein is called thyroglobulin. What is the name of this protein? Thyroglobulin. Like other proteins, it must be a chain of amino acids, right? There are many types of amino acids which are incorporated into this, but the point is the many amino acids, but one very important amino acid is tyrosine, right? Then there may be another tyrosine, then there are some other amino acids, and again there is a tyrosine. Of course, this tyrosine is like a tire mean it is like a ring of carbon. Then again there may be tyrosine residue. So what we really see that thyroid, these follicular cells are activating such genes which make such type of messenger RNA, that when these messenger RNAs are translated, they eventually end up in formation of a protein, and this protein is called, yes, thyroglobulin. Of course, this protein, once it is synthesized, right, once this thyroglobulin protein is synthesized, one thyroglobulin protein has many tyrosine residues. It has many tyrosine residues, right? This protein will be pumped in endoplasmic reticulum, right? And once this protein is going into endoplasmic reticulum, right, when this protein will enter into um, endoplasmic reticulum, it will get there glycosylated. So it will convert into glycoprotein. From there, from the endoplasmic reticulum, this will be packed into small vesicles, and through those vesicles, it will be released towards the, moving towards the, yes, apical side. On the way, it has to be finally packaged through which operation? What is the structure? Golgi apparatus. Again, let me tell you, step number one here, gene is activated. Step number two, messenger RNA is synthesized. Step number three, messenger RNA translated into a protein. This protein is a very unique protein because in the amino acid sequence, a lot of tyrosine is incorporated into this structure. For example, this is a tyrosine, there may be a tyrosine, there may be a tyrosine, there may be a tyrosine. So we can say this protein is rich in tyrosine. This tyrosine rich protein is taken up by endoplasmic reticulum. There it is further modified and glycosylated. Then eventually it appears into what? Golgi apparatus. There it is finally packed, and eventually it is ready to be secreted from apical face of the cell, right? So we can say that through the apical face of the cell, right, protein which is packed into this particular vesicle, right, this vesicle will fuse with the apical membrane, and eventually this protein will be released in where? Into luminal area. Now let's suppose I show this protein here. This is that protein, right? As you know, it has many amino acid, different type of amino acids, right? The point which I want to highlight, that there may be many types of amino acids which are present over there, but the most important amino acids which are relevant with us right now are your, which one? T3, so tyrosines, which are important for you, tyrosines. Is that right? Now this amino acid is different, this amino acid is different, this is tyrosine, this is tyrosine, again this is tyrosine, here it is another tyrosine, some other amino acids, again you end up with tyrosine and another tyrosine. So what we really see in this whole protein structure, which is called thyroglobin, many tyrosine amino acids are incorporated, many tyrosine structures are incorporated. Now as you know that one function of, what was this enzyme? What was the name of this enzyme? Please tell me. Peroxidase. Which peroxidase? I'm not very good at drawing. Please, no one should mind it. Right? And don't think it has anything to do with anyone very near and dear to you. This is tyrosine peroxidase. So one function is, it is converting what? Iodides into iodine. Other function which is there is that once, what is the structure? Thyroglobin. Second function of this protein is, okay, I think it's not good. Let's make it as plain as possible, right? Some people may feel uncomfortable with this type of situation. Now, second function of this enzyme is that it will fuse this iodine with the tyrosine residues. It will fuse these iodine with tyrosine residues. Now, due to the second function, what really happens, that we say that tyrosine iodine, sorry, due to the second function, the iodine, which is inorganic compound, when it becomes associated with tyrosine, we say iodine is organified. Iodine is organified. So iodine may bind here, or at in this tyrosine, iodine is binding at 2 points. Now listen, when iodine is binding at one point in the tyrosine, we call it monoidotyrosine. What is this function called? Monoidotyrosine. So monoidotyrosine. On this tyrosine, the next tyrosine, we have added how many iodides? 2. So because 2 iodines are there, we call it diiodotyrosine. It means one another function of this particular a very important function of this particular enzyme was not only to convert iodides into iodine, but help the iodine to get organified by fusing with the tyrosine. So this is its second function. Is that right? That it has led to the, what was this function? Organification. And this was its first function. What was it? Yeah. Now we have seen two functions of this enzyme. One function is iodides into iodine. This is peroxidase function. Is that right? This enzyme is thyroid peroxidase. Second function was, it was incorporating iodine with tyrosine, and when it was incorporating iodine with tyrosine, it was leading to organification of iodine. Second function. And when it will repeat the same function with multiple thyroids present over there, multiple tyrosines present over there, right? Eventually what will happen, that multiple mono and diiodotyrosines are formed. For example, here is again maybe one iodine, then another iodine. Here's iodine. It means they have double iodinated. If this tyrosine is double iodinated, this is diiodotyrosine. This tyrosine residue is also double iodinated. It is also diiodotyrosine. Is that right? Here, let's suppose again there is only single iodination, and here there is double iodination. And again we'll repeat it. Here it is double iodination. Here is also double iodination. So it means that this enzyme has led to oxidation of iodine, iodides into iodine. It has also led to organification of iodine, and it has one more very important function. You know what is that? It helps these two adjacent iodinated tyrosine to fuse with each other. Right? How? This is another function. Very important function over here. What is the other function of this? Other function of this enzyme is it helps these two adjacent to fuse with, yes, to fuse with each other. And if they fuse with each other, this, it means they are making a couple. So this is called coupling reaction. It means monoiodotyrosine, and here it is diiodotyrosine, they are coupled with each other. And when they are coupled with each other, it means that within the structure of thyroglobin, first two iodine residues were iodinated, and after they were loaded with iodine, they are fused with each other, right? And if they are fused with each other, what really happens? Monoiodotyrosine fused with diiodotyrosine. When monoiodotyrosine, right, fuse with the diiodotyrosine, then both of them together make triiodotyrosine. Triiodotyrosine. Again, let's focus on it and concentrate this concept. What was going on at this point? That here it was, what was it? It was tyrosine residue. This was also a tyrosine residue. Is that right? And this first tyrosine residue, it was having one iodine, and second tyrosine residue, it was having how many iodine? Two iodines. If first was called monoiodotyrosine, second was called diiodotyrosine, right? And if they are fused together, right, then they are called triiodotyrosine. Similar reaction may occur in some other parts of thyroglobin also. Let's suppose focus on this point. Let's suppose focus on that point. Let me explain it with more clarity. Let's suppose there is another tyrosine residue here, and there is another tyrosine residue here, right? And let's suppose in this case this is double iodination, this is also double iodination. So naturally this residue should be called diiodotyrosine. This should also be called diiodotyrosine. Is that right? Now the point which we have to understand, that if this magical enzyme also lead to coupling of this, right? They are really very happy. You know why? They are going to, yes, couple. Do they look to you as a male or females? Yes. What do you think? They are both males or females? No, no. Please let me know. Is a male or females? Let's focus on that and see. Yeah. You want the male and a female, but they look like each other. So there are two females or two males. Okay, let's come like this. You're laughing. You don't want to reveal your unconscious to me. Okay, fine. So what I was talking about, actually, they are not male or female. They are just tyrosine loaded with iodines, right? Now it has diiodotyrosine. This is also diiodotyrosine. Again, this enzyme come into action. And if this enzyme is coming into action, right, and if it lead to coupling of that, right? If it leads to, what is this? Coupling of that. And if both of them are coupled, now dihydrotyrosine fused with another dihydrotyrosine. Now this unit should be called tetrahydrotyrosine. Now you see the beauty of understanding that this was one single thyroglobin molecule. And within the thyroglobin molecule, there were too many tyrosine residues. And these tyrosine residues were getting iodinated. And once they iodinated, initially they lead to the formation of monohydrotyrosine and dihydrotyrosine. Within this whole structure, there are many monohydrotyrosine and there are many dihydrotyrosine. Then what happens? This enzyme will lead to coupling reactions. Is that right? When this enzyme will start coupling reactions and stitching together, of course, biochemically, adjacent iodinated tyrosine residues. If monohydrotyrosine fused with dihydrotyrosine, you end up with formation of T3. And if dihydrotyrosine is fused with nearby, coupled with nearby dihydrotyrosine, you end up with a structure, combined structure called T4, thyroxine. It means T3 and T4 are formed within the structure of thyroglobin. Now look, we come over here. There are some more amino acids here. And again, what is this? Tyrosine and another tyrosine. These are two tyrosine. If I say it is only iodinated at one point and other one is iodinated at 2 points, then what is this? Yes, monohydrotyrosine, and this is dihydrotyrosine. Is it right? Now, let's suppose this enzyme ignore this unit and this mono and di are not coupled. Mono and di are not coupled. It's quite possible in your life you come up with many people who are never coupled. At least they claim that they have never coupled. Right? Now, we can come to again some more thyroglobin unit. Again, this is, what is this? This is again tyrosine residues. And let's suppose here is iodine, double iodination here, and here is also iodine. Now what is it? Dihydrotyrosine and again dihydrotyrosine. And if thyroid peroxidase couples them together, right, what will happen here? Formation of T4 thyroxine. It is T1 and this is T2, theoretically speaking. It means this is one peptide chain in which tyrosine residues were first attached with iodine, and there was formation of monohydrotyrosine and dihydrotyrosine. Then the many points they were coupled with each other, and due to coupling reaction again by the same enzyme, what was the result? Yes, T3 formation, T4 formation. So we can say T3 formation and T4 formation is within the molecule of thyroglobin. Is that right? So we can show this molecule in a more simplified way like this.